In the autumn of 1991, two hikers crossing the Ötztal Alps on the border between Austria and Italy found a body emerging from a glacier. It was not a recent death. The body was 5,300 years old, a Copper Age man now known as Ötzi the Iceman, one of the most studied ancient humans ever found. In his stomach, the last meal he ever ate was still preserved. Red deer, wild goat, Eichhornwheat, and brown bear. He had eaten brown bear meat 3 days before he died. Somewhere in those mountains, killed by an arrow through the shoulder, still unknown who shot him or why. The bear whose flesh he carried in his stomach had lived and died in the Alps at a time when Europe looked completely different from the Europe we know. The forests were older, denser, more continuous. The megafauna were still present in numbers we can barely imagine. And brown bears were everywhere. Today, fewer than 50 brown bears remain in the Alps. We think we understand the brown bear. It is the largest land predator in Europe. The animal whose image has decorated coats of arms and city flags from Berlin to Bern to Moscow. It is the original bear, the one behind Paddington and Winnie the Pooh and the constellation Ursa Major. It has lived beside us long enough that it has entered the deepest layer of our cultural imagination, and the story we tell about it, about where it came from, how it spread across the northern hemisphere, what happened to it during the ice ages, and what the population clinging to survival in the Cantabrian Mountains of Spain has to do with the grizzly bears of Yellowstone, has been wrong in ways that the ancient DNA has only recently begun to fully reveal. What the genomes show is stranger and more consequential than anyone expected. This is what the DNA actually shows. Before we get into it, hit that subscribe button. Every video here digs up the real origin of something you thought you knew. Let's begin. The textbook story of the brown bear placed it as a species of whole Arctic distribution, meaning it lived across the northern land masses of Europe, Asia, and North America in a more or less continuous population, with regional subspecies reflecting local variation. The species, Ursus arctos, was thought to have originated in Asia, spread westward into Europe, and eastward across the Bering Land Bridge into North America, where it became what we call the grizzly bear. During the Pleistocene, the different populations, the grizzlies of North America, the brown bears of Europe, the bears of Siberia, and the Kamchatka Peninsula, were treated as variations on a theme. Related populations with a shared, recent common ancestor and broadly similar genetic histories. The fossil records seem to support this. Brown bear fossils appear in Europe from around 500,000 years ago and in North America from roughly 50,000 years ago, consistent with a westward origin and eastward dispersal across Beringia at some point in the late Pleistocene. The ancient DNA said something far more complicated was true. The first disruption came from the polar bear. Polar bears and brown bears can interbreed and produce fertile offspring, a fact known from captivity and from occasional wild encounters. The question was how closely related they actually were, and when they had diverged. The genomic analysis published in the early 2010s, drawing on whole genome sequences from both species and from ancient specimens, produced a result that surprised virtually everyone in the field. Polar bears are not simply the closest relative of brown bears. In certain genomic analyses, the polar bears cluster inside the brown bear lineage rather than beside it. The polar bear appears to have emerged from within a specific brown bear population, most likely a population that became isolated in an Arctic refugium during a glacial period and underwent rapid adaptation to the marine environment. This alone was remarkable. But what followed was stranger still. When researchers began comparing the genomes of modern polar bears to modern brown bears from different geographic regions, they found that the brown bears most closely related to polar bears were not the brown bears of Siberia, which lived closest to the Arctic and might have been expected to be the ancestral population. They were the brown bears of the ABC Islands off the coast of southeastern Alaska. The ABC Islands are a group of islands in the Alexander Archipelago. The brown bears that live there, technically a distinct subspecies, have been isolated on those islands for thousands of years, and their mitochondrial DNA, the genetic material passed down through the maternal line, is more closely related to polar bears than to any other brown bear population on Earth. The implication is extraordinary. The maternal ancestor of all polar bears was an island-dwelling brown bear population from what is now coastal Alaska, not an Asian or Siberian population as geography might suggest. At some point during the Pleistocene, a population of brown bears closely related to the ABC island bears became isolated in an Arctic environment and gave rise to the entire polar bear lineage. But here the genomic story bifurcates into something even more complex, because the mitochondrial DNA, which traces the maternal line, says one thing. The nuclear DNA, which traces both maternal and paternal lineages, says something partially different. The nuclear genome of polar bears shows significant contribution from brown bears at multiple points in their history, suggesting that after the initial divergence of the polar bear lineage, there were repeated episodes of hybridization between polar bears and brown bears, mixing genetic material in both directions. The polar bear is not a clean derivative of one brown bear population. It is a mosaic, assembled through initial isolation and repeated secondary contact across the Pleistocene. The second revelation concerned Europe, and it overturned the idea of a simple, uniform European brown bear population more completely than any previous analysis. When ancient DNA researchers extracted and sequenced genomes from brown bear bones recovered from cave sites across Europe, spanning the last 50,000 years, they found a population structure of astonishing complexity. European brown bears during the Pleistocene were not one population. They were multiple genetically distinct lineages, occupying different parts of the continent, separated by the barriers of glaciers and unsuitable habitat during glacial maxima, diverging over tens of thousands of years in their respective refugia. During the coldest phases of the last ice age, when ice sheets covered northern Europe and open steppe replaced much of what is now forest, brown bears retreated into isolated refugia. The bears of each refugium diverged from the bears of the others. The Iberian Peninsula harbored one population. The Italian Peninsula and the Balkans harbored others. The Caucasus harbored another, genetically distinct from all the Western European lineages. When the ice retreated and the forest expanded northward, these refugial populations expanded too, recolonizing the newly available landscape, and as they did, they met. The genetic data from ancient specimens shows a complex pattern of admixture across post-glacial Europe, different refugial lineages moving into the same regions, hybridizing along contact zones, producing the genetically complex population that historical European brown bears actually were. But then the ancient genomes revealed something that the modern genomes alone could not show, something that had been lost. The ancient bear population of Ireland stands as one of the most striking examples of cryptic lineage extinction in the Pleistocene megafauna record. The bears that lived in Ireland during the last ice age, whose bones have been recovered from cave sites across the island, were not the ancestors of any modern bears. They were a genetically distinct lineage that was wiped out, replaced by bears that colonized Ireland the glacial period from a different source population. The Irish cave bears left no descendants. Their entire lineage is extinct. The only record of their existence in the ancient DNA extracted from their bones. This pattern recurred across Europe. The ancient genomes showed that the lineage composition of European brown bears before and after the last glacial maximum was not the same. Certain ancient lineages disappeared, others expanded to fill the space. The modern European brown bear population, diverse as it is, represents only a subset of the genetic diversity that existed before the last ice age. The third revelation came from the genetics of the living populations, and it is the one with the most painful contemporary implications. When the genomes of modern European brown bears were analyzed across the continent's surviving populations, researchers found a pattern of fragmentation so severe that it amounted to a genetic catastrophe in slow motion. The brown bears of Scandinavia, the Carpathians, the Dinaric Alps, the Balkans, the Cantabrian Mountains of Spain, the Pyrenees, the Apennines of Italy, and the scattered remnant populations of Eastern Europe, not a single population. They are isolated fragments, separated by centuries of habitat destruction, hunting, and human settlement, each fragment carrying a reduced subset of the genetic diversity their ancestors held. The Cantabrian population in northern Spain is particularly instructive. These bears, roughly 250 animals, split into two subpopulations barely in contact with each other, are the last wild brown bears of the Iberian Peninsula. Their genomes told a story of repeated bottlenecks, first from the natural fragmentation of the glacial refugia period, then from centuries of human persecution, then from 20th century hunting that reduced the population to perhaps 50 animals in the 1990s. The genomic data showed that the Cantabrian bears carry a very high load of deleterious mutations, variants that in a larger population would be eliminated by natural selection, but that in a small inbred population accumulate and become fixed. The bears are also showing signs of inbreeding depression in reproductive success, measurable in reduced litter sizes and juvenile survival rates compared to healthier populations, and then the ancient DNA provided a comparison that made the loss visible. The Cantabrian bears of today carry a fraction of the genetic diversity that bears in the same geographic region carried 20,000 years ago. The ancient bears of the Iberian refugium were genetically rich, diverse, a population with a full range of variation that tens of thousands of years of isolation and adaptation had produced. What remains today is the depleted remnant, not just in numbers, but in genetic content. The fourth revelation is the one that connects back to Ötzi and his last meal, and it concerns what happened to European brown bears in the period between the end of the Ice Age and the present. The genomic analysis of museum specimens and archaeological bones allows researchers to track the genetic composition of brown bear populations through historical time, not just prehistoric time, and what this historical genomic record shows is a progressive homogenization and impoverishment of European brown bear genetics that tracks precisely with the expansion of human settlement and the intensification of persecution. Medieval bears in Germany and France show higher genetic diversity than bears in the same regions just a few centuries later. When hunting pressure intensified and populations began to fragment, the bears that survived into the 19th and 20th centuries in the remnant mountain strongholds were already impoverished compared to their medieval ancestors, who were themselves impoverished compared to the post-glacial population. Each wave of human pressure left a visible mark in the genome. The loss of a forest here, the intensification of hunting there, the bounty system that paid for bear deaths across 19th century Europe. Each event narrowed the gene pool a little further, until what remained in places like the Cantabrian Mountains and the Apennines of Italy were tiny populations carrying the genetic legacy of centuries of attrition. The fifth revelation came from a comparison that nobody had initially expected to be as informative as it turned out to be, between European brown bears and North American grizzly bears. When whole genome sequences from brown bears across the global range of the species were compared, the pattern of genetic diversity was not what the simple out-of-Asia dispersal model predicted. North American grizzly bears, despite being geographically distant from the presumed Asian origin of the species, showed higher genetic diversity in certain genomic regions than many European populations. This was unexpected. A population that had dispersed furthest from its origin should show reduced diversity through the founder effects of dispersal. The explanation that emerged from the data was sobering. North American grizzly bears have, in most parts of their surviving range, suffered less extreme population fragmentation and less severe historical bottlenecks than European brown bears in the 20th century. The comparison exposed not a difference in evolutionary history, but a difference in the degree to which recent human persecution had degraded genetic diversity. European brown bears have been more thoroughly devastated by recent human action than the genomic baseline of their species warrants. The final layer of the genomic story is the one that the reintroduction programs across Europe have had to grapple with most. directly. When conservation managers in the 1990s and 2000s began augmenting the tiny remnant bear populations of the Alps and the Cantabrian Mountains with bears from outside populations to rescue them from inbreeding depression, they faced a choice that the ancient genomic data made harder. Which source population to use? Bears are genetically structured. Using bears from the wrong population could introduce inappropriate gene combinations, disrupt local adaptations, produce hybrids that were less well fitted to local conditions than either parental population. In the Alps, the augmentation was done with bears from Slovenia, the Dinaric population, genetically different from the last original alpine bears. The program was controversial. Locals objected. The bears killed livestock. Some were killed illegally, and the genetic data showed that the introduced bears and their descendants were indeed genetically distinct from the historical alpine population. The ancient DNA added context that complicated the policy debate further. It showed that the historical alpine population was itself a complex mixture of different post-glacial lineages, not a pure distinct strain that needed to be replicated exactly. The Alps had been recolonized after the Ice Age by bears from multiple refugial sources. The idea of a single authentic alpine bear genotype was, the ancient data suggested, a fiction. Bears had always mixed across the Alps as populations expanded and contracted. What was happening now was not genetic contamination of a pure lineage. It was, in geological terms, another episode in a long history of range shifts and population mixing. The ancient DNA did not resolve the policy debate. It never does. But it changed the terms on which the debate was conducted, introducing a deep time perspective that the conservation argument had previously lacked. Today, the brown bear of Europe exists in a situation that has no real historical precedent. Small populations, intensely monitored, their genomes sequenced, their movements tracked, their kills documented, their politics managed. They are simultaneously among the most studied animals in the world and among the most endangered in Europe. The bear that c8 5,300 years ago in the Alps was part of a population that stretched from Spain to Siberia without a gap. The animal was everywhere. It was in the forests of England and Scandinavia and Poland and Romania and Greece, and it was in the mountains of Turkey and Iran, and it crossed Beringia into North America, where it became what we call the grizzly. Its genome carried the accumulated genetic wealth of hundreds of thousands of years of adaptation across the whole Arctic. What remains in the Alps today is 50 animals. What remains in the Cantabrian Mountains is 2. So populations of 250, barely in contact, carrying a genome that the ancient data shows has lost a significant fraction of the diversity it held 20,000 years ago. On an autumn morning somewhere in the Cantabrian Mountains of northern Spain, a brown bear is moving through dense oak scrub, methodically eating acorns before the first frost. She is a large female, perhaps 15 years old. She has raised 5 cubs in her life. Her genome carries within it a record of everything her lineage has survived. The Pleistocene refugium, the post-glacial expansion, the medieval forests, the bounty hunters of the 19th century, the final compression of the 20th. She does not know any of this. She knows the acorns. She knows the frost coming. But her genome knows all of it, and we are only now learning to read what it contains. The DNA is only beginning to tell us what it knows. If you found this story remarkable, consider subscribing. There are more origins to uncover, and the DNA is only beginning to tell us what it knows.